Today's topic is regulation of eukaryotic gene transcription. So we looked at positive and negative regulation of transcription of operons in prokaryotic systems, but regulation of eukaryotic gene transcription is far more complex. And it's more complex because if you think about it, if you think about eukaryotic cells and eukaryotic organisms, take a human, for example, we have 20,000 plus different genes, but our cells do so many different things. So let's kind of just go back, way back to the original sort of situation where the egg and the sperm meet, the 23 chromosomes here get together with the 23 chromosomes in the sperm. When fertilization occurs, when one of these lucky sperm penetrate through the membrane, then all other sperm are cut off, and we have a complete 46 total chromosome composition. That triggers development. And at that point, we have what's called a zygote. So this is a fertilized egg, a zygote that undergoes mitosis, divides one cell, becomes 2 cells, 4 cells, 8 cells, 16, etc. These early stages, these cells are undifferentiated. They're unspecialized. They're stem cells. The zygote in particular can become any cell type in the entire body, right? But as time goes on, as we get 3 or so days after fertilization, then the cells begin to differentiate. Notice that these ones on the exterior of the cell mass begin to change. It's depicted here in blue. A cavity begins to form, and here we have the blastocyst stage. These are if you've heard of the term embryonic stem cells, this is the stage that they're referring to. And these can become most of the tissues in the body. They begin to differentiate into the different layers. And some will become connective tissue, some will become organs, etc. Those cells, as they change, they still have the same genes. So how can they become so different? And if you think about a human, how many different cell types do humans have? We've got red blood cells, neurons, heart cells, skin cells, liver cells. Depending on how you classify different cell types, we can say humans have around 200 different cell types, meaning those cells are doing different things. They have different specialized function. But they have the same genes, they have the same information, so how do they act and do such very different things? And that has to do with what genes are being expressed. And so let's take a look before we dive into eukaryotic gene expression. Let's look at the major differences from prokaryotic. And some of these were mentioned before, but it won't hurt to look at them again. First of all, eukaryotic genes are not polycistronic. All genes are monocistronic in eukaryotes, meaning you have one gene coding region per promoter. Nothing's transcribed together. So here's a polycistronic, which you might see in prokaryotes, but in eukaryotes, you've got a single protein coding region per RNA transcript. Generally, eukaryotic gene transcription are positively regulated. So positive regulation is the primary mode of transcription regulation. And remember that the proteins that mediate positive regulation are generically referred to as activator proteins. And prokaryotic systems tend to be negatively regulated with repressor systems. Another key difference is that the degree of expression or the degree of a gene being turned on varies. So when we talk about turning a gene on, as I said before, a better word is upregulated. Upregulation in eukaryotes is in the range of 2 to 10-fold increase in expression. Okay. Whereas in prokaryotes, it's far more drastic. It's more like 100 to 1,000-fold increased. So there's more subtle changes in expression in eukaryotes as opposed to, prokaryotes where it just blasts the gene expression. If you look here too, here's our bacterial on the left, here's our eukaryotic. In eukaryotic cells, we have to deal with chromatin. So eukaryotic genes are physically less accessible. Let me put that down here. That means that to get to the naked DNA, you actually have to unravel the chromatin to get to naked DNA. And another key difference is that in prokaryotes, most of the regulation occurs at the level of transcription. Either it's transcribed or it's not transcribed. It's turned on or it's turned off. Eukaryotic gene transcription is regulated at a number of different levels. So eukaryotic gene expression, I should say, going from the gene to a functional protein is regulated at every stage. And that's going from the DNA transcript at the level of the RNA and at the level of the protein translation itself. And we'll even find out that access to the DNA out here is a level of regulation. So I'll keep continuing to go back to this figure where we've got our chromatin. So one level of regulation is going to be at access to the DNA. Once we have access to the DNA, then we can control gene expression by controlling transcription. The processing of the messenger RNA can be controlled in the half-life of the messenger RNA, as well as the translation process and even at the level of the protein itself. So let's start up at the top. Regulating access to the DNA. And this is generically referred to as epigenetic regulation. And it's going to regulate whether or not transcription can occur. The term epigenetic means above the genome. And so this is independent of the gene sequence. So it's modifications that occur above the genome, above the sequence of the A's, T's, C's, and G's. And there's a couple of different major types. There's cytosine methylation, so there's changes to the DNA itself, and then there's changes to the histone protein. So we're going to look at both of those, and we start with cytosine methylation. There's a lot of information in these words. Cytosine, you know what this is, right? It's the C nucleotide. Methylation, little CH3 group. So cytosine methylation is the addition of a methyl group to cytosine nucleotides in DNA. And it's specifically, it's a methyl group that gets added to carbon 5 of the cytosine base. Let's see. Here's cytosine. This is just the nitrogenous base component of cytosine. It's a single ring structure. It's a pyrimidine. This is the normal structure. An enzyme called DNA methyltransfer comes in and pairs carbon 5. This is a methylated cytosine. So the process of this is that a DNA methylase alternatively called a DNA methyltransferase, is an enzyme that adds the methyl group to carbon 5. This happens in regions, even though it's above the genome and it's independent of the unique DNA sequence, it often occurs at CG-rich regions, which we call CPG-rich islands. And so all this means is that it's sequences that are heavily enriched in CG sequences, and that's in 5' to CG. So many of these in a sequence. It doesn't have to be exclusive, but it means it's rich in CGs. And then when you have, so let's imagine here, let's go CG, 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 CG. We'll get an A in there. It doesn't have to all be CGs. But if this region, if this CPG-rich island is heavily methylated, which I'm showing here with little asterisks, heavily methylated DNA generally has reduced transcription. So in general, heavily methylated DNA has reduced transcription. These CPG-rich islands are usually located near regulatory sequences, promoters, and other regions that are involved in regulated transcription. So, and if there's heavy methylation of the Cs, then that results in sort of reduced transcription. And I like to think about it this way. If you think about the proteins that are going to bind to the DNA to promote transcription, essentially, if it recognizes C's and G's, this is like a little disguise. You know, maybe think of it as like an invisibility cloak or something, right? It disguises this regulatory region, maybe promoter region. And so those proteins that would otherwise turn on transcription don't recognize and bind to this region as efficiently, so it results in reduced transcription. Here's another depiction here. Here's a CPG island. This is a regulatory region in orange. We haven't discussed this yet. Here's a promoter. In the case of this activator protein binding to this enhancer, if it's not methylated, it can bind and it can turn on transcription. Whereas if it's heavily methylated, the activator protein doesn't recognize the sequence and therefore does not bind and transcription is not turned on. Here's another depiction showing here again that this is at the level of DNA where these changes occur. And notice that if it's CG on one strand, on the complementary strand, it is the same sequence CG 5' to 3'. This methylation pattern can be inherited or is inherited, or you can think of it as maintained during cell division. Well, how is that possible? Well, let's think about it. What happens during cell division? Well, the DNA has to replicate. Here's our parent strands, they separate, so parent. Here's our newly synthesized strand. Now this does not have methylation on it right now, right? Parent with the methylation. Here's a new strand without it. We call this hemimethylated DNA. So during replication, DNA is hemimethylated, meaning one strand is methylated, the other is not. The strand that's methylated is the parental strand. And that actually serves as sort of information. This methylated parent is recognized by those methyltransferases and it serves as a template to methylate the new strand. So in that way, we can keep the methylation pattern. We call this maintenance methylation. So the methylation pattern is maintained with cell divisions because this parent strand contains information to direct the new methylation of the newly synthesized strand. A special type of cytosine methylation that occurs is called sex-specific genomic imprinting, or sometimes just genomic imprinting. So this is a special type of cytosine methylation, and it is a differential methylation of the cytosines in eggs versus sperm, in the male versus the female gametes. Sex specific, meaning that in the male gamete, the sperm, a set of genes are methylated, whereas those would not be methylated in the female, and then vice versa. Not all genes, but if we know of currently a few hundred. We know of a few hundred genes that are sex-specifically imprinted. In the germline formation, during gamete formation, the methylation pattern is erased. So the methyl groups are taken off the cytosines. On both chromosomes, and the methyl pattern, so the methyl pattern is reformed based on the gamete of the sex. So it's reestablished in a male or female specific way. So let's look at, this is best seen by example. So we'll use an example of a region of chromosome 15. At 15q11, there's several sex-specific imprinted genes. And so two of those genes at 15q11 are called SNRPN and nectin, and UBE3. So we'll look at 3 genes. These 3 genes are sex-specifically imprinted. So they're one of these few hundred different genes that are methylated during the gamete formation. So in a female-derived chromosome, and remember, each individual has a chromosome that was derived from the female lineage and the other from the male. So in the female chromosome, let's kind of depict this. Ooh, sorry. Oh, that's a horrible looking piece of DNA. But bear with me here, we'll say S and U. These 3 genes are in the proximity of chromosome, the long arm of chromosome 15, region 11. In the female derived, in the maternally derived chromosome, the snerpin and the nectin are methylated. The snerpin and the nectin are methylated, which means they're not expressed, but the UBE3 is not. So in the maternal snerpin, the nectin are heavily methylated, meaning they're not transcribed in the maternal chromosome. UBE3 is not. That means that all the UBE3 that's derived in human cells is derived from the maternal chromosome. So can you guess what the male pattern is then? So your paternal derived chromosome has the same genes in the same region, unless there's some abnormality. But these are genomically imprinted during germline development, and in the paternal lineage, in the sperm, the chromosome that you inherited from the male parent, the UBE3 is methylated and is therefore not expressed. Snerpin and nectin, however, let's take a look at that. That would be a little bit different. They are expressed. So essentially, these genes, these sex-specifically derived genes are expressed really only in one copy. And they're expressed either from the paternal or the maternal chromosome, not from both. Here again, we can look at a visual depiction, chromosome 11, long arm, region 11. Here are, like I said, there are several genes here, but here's UBE3. Here's snerpin. Let's see, that might be named something different. These are in here, and it shows you which methylation pattern exists. So in red are the maternally expressed genes, and in purple are the paternally expressed genes. So these are almost similar to like X-linked genes, right? Because you only have the expression of one copy of these. There's also a health related phenomenon with this. Imagine, what if you had a deletion in this region? So what if you had a 15q11 deletion? That means you would lose those genes that are in that region, and they only exist essentially for expression purposes in one copy. So it depends on whether or not it's a deletion in the maternal chromosome or in the paternal chromosome. So if there was a 15q11 deletion in the maternal chromosome, so we're missing, make this like we have a deletion here, but we have the paternal chromosome. What does that mean sort of at a molecular basis? Well, if we remember, the maternal chromosome is where UBE3 expression is. This is heavily methylated in the paternal chromosome, right? So essentially, if we have this deleted in the maternal chromosome, then we're not going to have expression of UBE3. So we would have no or significantly reduced. And UBE3 is important for for development, for human development. So the result of this is a condition called Angelman syndrome. Angelman syndrome, it has a frequency in the human population of 1 in 15,000-ish live births. The phenotype of Angelman syndrome, there's moderate to severe intellectual impairment. there's very often no language development. So people with Angelman tend not to be verbal, and their facial features often show sort of a smile, laughter sort of depiction. I've got a picture of a child with Angelman here. So this is a girl with Angelman syndrome, and there's sometimes hand flapping and stuff as well. But what if you had a deletion? What if you had a 15q11 deletion from the paternal lineage? Well, then we have the opposite situation where the maternal genes are the only ones that are there. And so if you remember what we have here, serpin and nectin are heavily methylated. Our paternal chromosome is missing these genes. Sorry, this was a mistake here. This is the maternal chromosome. Our paternal chromosome is missing these genes. So in this case, we have no serpin and nectin, with significant reduction in serpin and nectin, and that also has a phenotypic result. The result of this is not Angelman syndrome, it's Prader-Willi. Prader-Willi syndrome also has a frequency of 1 in 15,000-ish. The major phenotype of Prader-Willi syndrome is insatiable appetite. Insatiable appetite and a resulting morbid obesity if there's not severe sort of social structured environment for food access. There can be some mild developmental delays, but they're fully verbal. An individual with Prader-Willi, if they have any intellectual impairment, it's very mild. There can be some behavior issues because they are hungry all the time. So here's an individual with Prader-Willi syndrome. So these are two very distinct conditions, but they're both due to a 15q11 deletion, right? So it depends which deletion you inherited. If you inherit the paternal deletion, it results in Prader-Willi. So if you inherited a deletion from the paternal chromosome, the one inherited from dad, it results in Prader-Willi. Whereas if you inherit the deletion of the maternal chromosome from mom, it results in Angelman syndrome. So I said there were two major types of epigenetic regulation of gene transcription. So we've discussed cytosine methylation, and we've discussed sort of the special sex-specific imprint that it is kind of, like I said, differential cytosine methylation. The next type of epigenetic modification are histone modifications. Histones should ring a bell. We talked about them before. They're a component of the nucleosome, right? So here's a nucleosome. And if you remember, the nucleosome is DNA wrapped around an octamer, a histone. In that histone octamer, there are two each of histone H2A, H2B, H3, and H4. So here you see them. They're very similar, but they are different. They're coded for by different genes. And if we look a little more closely at these histones now, each histone has an N-terminal tail. Which you can see here. These tails are unordered, as opposed to this globular structure, which is folded into its three-dimensional form, which is fairly stable. These tails are flexible, moving. They are unordered. They don't have secondary structure. They don't have tertiary structure. They're kind of just, think of a tail wagging around, right? So histones have these N-terminal tails that are unordered. Put that in here, unordered. And they're rich in lysines. Now you might not remember this, but what is the chemical characteristic of lysines? Lysines is one of the 3 amino acids which are positively charged. So the R group of lysine residues are positively charged. Now let's think about what that means. So if we've got a bunch of positive charges on here, can we see where this might be going? And these things can move around. Well, what if positives are attracted to? Positive charges are attracted to negative charges. Can we think of any molecules that might be negatively charged? Yes, this phosphate backbone is very negatively charged. So these tails interact when they're unmodified. So unmodified tails interact with the DNA to keep this nucleus, condensed, right? To kind of lock it into place. So it interacts and it locks it into place. So that's the premise now of modification of histones. So if we add functional groups to these lysine residues in the histone tails, we can change how the lysine residues interact with the DNA. And so there are different ways. Oh, here we can look at our condensed chromatin, but we can modify the histones and open up the chromatin, and some modifications actually cause it to wrap up more tightly. Common modifications is acetylation, histone acetylation. This is an addition of an acetyl group, which looks a little something like this. To the lysine residues in the histone tails. Well, remember those tails normally are positively charged and they're interacting with the DNA, keeping things nice and condensed. Here is a cartoon depiction of an acetyl group. It dampens that positive charge. It helps to pop open the chromatin. It helps to decondense the chromatin. So, and specifically, the acetyl groups are added to H3, histones H3 and H4. And it results in the DNA being more loosely packaged or opened. Let's say more accessible. And it's more accessible because it allows the chromatin to decondense. Now there are 2 enzymes that I do want to point out to you, because if you pick up some scientific literature, you might see these. Epigenetics is a huge area of research. It's newer. It's only been around, well studied or studied in maybe the last 20 years. And so HATs are enzymes called histone acetylases. And HATs add the acetyl groups. To H3 or H4. If we add them, there also has to be a mechanism to remove those, and those are HDACs or histone deacetylases. And deacetylases remove the acetyl group and allow the chromatin to recondense. So again, if we go back here, HATs add acetyl groups, pop open the DNA, whereas HDACs remove them and allow for the chromatin to be condensed. Now, acetyl groups are not the only things that can be added to the histones. There are other types of histone modifications. So now we're zooming in, kind of looking at the residues in histone tails. Here we see H3 and H4, actually H2A and 2B are over here as well, but let's focus on 3 and 4 because those are the ones we talked about. In green are positions where acetyl groups can be added to. K is the one letter code for lysine. So K, and where you see K, that's a lysine residue. This is arginine, this is serine, but mostly notice how there's a lot of Ks. And you can see that this green marks mean they're active marks, mean they open up the DNA. But we can also have inactive additions to those. So another type of functional group that can be added to the histone tails, and some have multiples. So some of these residues is another name for just amino acid. So this would be residue 36, residue 27, have multiple modifications that occur. So in addition to histone acetylation, there's histone methylation, not to be confused with cytosine methylation. The methyl group is the same, but where it's being added is different. So methylation of histones is more complex. Some methylations in here, in aqua, promote the DNA being more accessible, whereas other positions of methylation promote it being less accessible. So methylation of histones has either promotes either DNA being more accessible or less accessible depending on where those methyl groups are added and how many methyl groups are there. And there's one final, let's see, can we find, oh, here we go. P, guess what that is? We can also have histone phosphorylation. Now you know what a phosphate group is like, right? That's got negative charges, negative charges. How do you think that negative charges in these histone tails is going to interact with the negatively charged DNA? It's going to be repulsive. So these phosphorylation events tend to make the DNA more accessible. So what you find is in different types of cells and different cell types, you have these different patterns of histone modifications. Patterns of histone modifications and patterns of cytosine methylations. So different tissue types have different patterns. It's not completely understood how those patterns are established, erased, and reestablished, right? We do know that there are patterns that are specific to different cell types. We know the major enzymes that are involved, but how does that whole process occur? It's not been fully elucidated, but it's a really active area of research. So let's watch this.
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world. such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
So the final thing I want to say about epigenetic regulation is what was just said in that video, that we're learning, and again, it's a really hot area of research, that environmental factors, environmental exposures to things like toxins, cigarette smoke, foods, stress, as was mentioned there, that different exposures to things in the environment can alter the epigenetic modification patterns. And some of these alterations in the epigenetic modification patterns are also correlated with disease. So I'm just going to put down here, this is a hot area of research.